Hi there, and welcome to this video on A-level biology for the AQA specification, focusing on the topic of organisms exchanging substances with their environment, and in particular, on digestion and absorption. I'm Manisha from StudyMind, where we help you to revise A-level biology with our helpful video tutorials tailored to your subject, your specification, and to you. If you're new here, please make sure to click that subscribe button. And whilst you're watching, feel free to leave any comments down below of anything you're unsure about, and let us know if it's your first time watching so we can send you our free revision resources. We also have helpful timestamps to guide you through the specification. So, let's get started. Welcome back to Lesson 3. Now we will look at the small intestine and absorption. Our next specification point is to look at absorbing the digested products. The small intestine is effectively a very long tube. The outer walls of the tube are lined with smooth muscle. The inner wall, which faces the lumen, which is the hollow part through which digested materials pass through, is lined with millions of epithelial cells. The inner wall of the small intestine has many folds called villi, which are designed to increase its surface area for absorption. This will also promote increased absorption of nutrients. The cells which line the villi also have small folds on their surfaces, known as microvilli. This will increase the surface area further. Each villus has capillaries and lacteal vessels located underneath it. The purpose is to absorb the nutrients that are absorbed by the epithelial cells of the villus. The blood capillaries will connect to the hepatic portal vein, and the lacteal vessels will connect to the lymphatic system. We can see this here. The absorption of products of digestion begins in the duodenum. The majority of the absorption, however, will occur in the ileum. Specifically, the absorption occurs at the microvilli surface of the epithelial cells. Next, we will look at the co-transport mechanisms for absorption. This is a summary of the route of nutrients. First, the nutrients are absorbed from the lumen of the ileum through the villi. Next, they reach the epithelial cells of the small intestine. They will then reach the bloodstream to go into the portal vein and the lymphatic system. The diagram here is to give you a visual representation of what we've just explained. Monosaccharides are absorbed by the epithelial cells lining each villus. Glucose travels from the lumen of the ileum to the bloodstream through the sodium-glucose co-transport mechanism. This mechanism uses a co-transporter proteins, and active transport is required to pump the sodium ions out of the epithelial cells and into the bloodstream. Let's quickly recap what we learnt in a previous video. There is a sodium-potassium pump on the basolateral membrane. This will actively transport the sodium ions out of the epithelial cell and into the bloodstream. Next, the sodium concentration in the epithelial cell will fall, compared to the ileum. Then, sodium and glucose can enter the cell via facilitated diffusion. This occurs down a concentration gradient. Sodium and glucose then diffuse out of the basolateral membrane into the blood. Water will follow through aquaporins. Here's a quick summary of all the steps. Now let's look at the absorption of monosaccharides. Fructose is absorbed via facilitated diffusion. This involves a different carrier protein to glucose transport. Here's a diagram of the facilitated diffusion. Amino acids are absorbed through the epithelial cells of the villi. There is active transport of sodium from the epithelial cell to the lumen of the ileum through the apical membrane. The sodium will diffuse back down its concentration gradient, carrying amino acids via co-transport. Then, the amino acids can be transported into the bloodstream. In summary, sodium is actively pumped out into the lumen. It can diffuse back in alongside an amino acid, which is co-transport. We already covered glucose transport in a previous tutorial, so feel free to re-watch it again if you'd like some more detail. Finally, we will look at the role of micelles. Absorption of glycerides and fatty acids requires the formation of micelles. Micelles are tiny structures that are formed when a bile salt combines with an undigested glyceride and fatty acid. Micelles are transported to the epithelial cells lining the villi in the small intestine. Then, they break down near the villi, and their contents can enter the epithelial cells, as shown here. Now, the smooth ER can reform the triglycerides, using the absorbed monoglycerides and fatty acids. The triglycerides are transported to the Golgi apparatus. This will then package the triglycerides into structures known as chylomicrons. These will pass out of the cells of the villus and into the surrounding tissue fluid via exocytosis. The exocytosed chylomicrons will enter into the lacteal vessels and circulate in the lymphatic system. They can enter the circulatory system by draining into the left subclavian vein from the thoracic duct of the lymphatic system. So just to recap, the micelles are transported to the epithelial cells, and then they break down near the villi, which will release their contents. Next, the contents will enter the epithelial cells, and the released contents diffuse into them. The triglycerides are reformed by the smooth ER. Then, the Golgi will package the triglycerides, which can pass out of the cells of the villus. Then, the chylomicrons will travel into the lymphatic system, and they can enter into it via the left subclavian vein. Here's another summary for your reference. It's really important you know this for your exams, so make sure you learn these 6 steps well. We've now covered all the learning objectives for today's lesson. Feel free to skip back through the video and the previous one to recap anything you feel unsure about. We've now completed Lesson 3. If you enjoyed this tutorial, make sure to subscribe by clicking down below and leaving a comment of the topic that you'd like to see a video on. Click here to watch the rest of our videos in our A-Level Biology series, or visit our website, studymind.co.uk, for past paper compilations by topic and specification.